Hello again, everybody. Welcome back to this series of videos where I'm going through the AP progress checks on AP Classroom um, and kind of working my way through all the FRQ questions to help you guys get ready for the online exam that's coming up. So this question is from AP Unit 4, which is cell cycle and cell signaling. So it sort of straddles the two of our units. It was kind of the very end of our biochemistry, cell biology, and sort of the very beginning of our genetics unit. And as you'll see, this one is more about cell signaling, and the second question in this unit is more about cell cycle. So this kind of straddles our two units, Unit 2 and Unit 3. But everything involved we've covered. So let's take a look at question 1 here. So growth hormone and insulin are protein hormones that regulate carbohydrate metabolism by hepatocytes, liver cells, through the activation of intracellular signaling pathways. Researchers investigated whether two hormones trigger the same or different intercellular signaling pathways in hepatocytes. The researchers added either growth hormone or insulin to parallel cultures of hepatocytes. At several time points after the hormone addition, they purified total intracellular protein from the cells and quantified the level of the phosphorylated forms of two proteins involved in two different signaling pathways. Figure 1a and figure 1b. Okay, so let's take a look at this. There's a lot of data there. Okay, so again, all right, describe the first interaction that triggers cell signaling to begin in a target cell. Okay, well, this actually doesn't involve any of the data. This is just kind of the basics of what we learned when we looked at Chapter 11, which was cell signaling. And you guys should all know that a signaling molecule, aka a ligand, binds to a receptor. Whether it's inside the cell or outside the cell depends on whether it's, a polar hormone like epinephrine or a non-polar hormone that can go through the cell membrane, like a steroid, like testosterone or estrogen, will bind to a receptor in the cytoplasm. But either way, there's a signaling molecule, aka a ligand that's going to bind to a receptor and promote a cellular response. So just kind of very basic stuff. So let's take a look at part B. Here's where we start to dig into the data a little bit. So identify a dependent variable in the experimental design. Okay, fairly easy. So all you're looking, as you remember, the independent variable is going to be what you are changing, right? So it looks to me like they are changing the type of hormone, insulin or growth hormone. That's going to be your independent. And then the dependent is going to be what the researchers or what you are measuring, and quantify the level of phosphorylated forms of two proteins, right? So the dependent variable, okay, is what they measured. Right, that's the measured variable. And in this case, as we said, that was going to be phosphorylated signaling protein, right? So phosphorylated signaling proteins. Okay, levels. Cool. All right, so let's look at the second part here. Negative control. Okay, negative control for the effects of hormone addition. So this has come up before. So a negative control is basically something that's not there, right? Basically, the independent variable is not present. And so in this case, we already kind of were thinking about what the independent variable was, which was the hormone. So your negative control would be no hormone present, because your negative control is just whatever you're measuring is not there. Okay, so, and then identify a likely reason why the researchers continued their experiment for 240 minutes. full disclosure, I do not care for this question. I was really confused when I first started to look at it. I guess the most obvious answer, what I probably would have gone with, is they just wanted to see if anything happened over a long timeline. Um, I don't think there's any real specific content that they really wanted. Just sort of to see the long-term effects. And I suppose that, hormones can be fast-acting like epinephrine, or they could have long-term effects like a steroid hormone like testosterone. But yeah, I don't think there's any real content that they're testing there. I guess you just want to see, like, I guess if there was like another spike there, or if this started to go slowly and then crashes. I don't know. That's the best I can do. So yeah, looking at the scoring guidelines, ligand, chemical messenger, binds to a receptor protein, very dependent variable, phosphorylated signaling protein, negative control, a 0 time point when hormone is added, or adding no hormone or a denatured hormone. So again, independent variable, not a factor. And then hormone might take some time to start, or they might have wanted to see how long a response to the hormone lasted. Sure, so effects over a long period of time. Again, if there's specific content they were looking for there, I don't see it. Doesn't mean it's not there, but I don't see anything specific that they were looking for. Okay, so let's look at part C. Okay, so describe the hormone and signaling protein combination that produce the greatest and most prolonged response. Okay, pretty simple so far. Clearly, you can see that the insulin, right? The insulin definitely produced a longer and prolonged response just by looking at the data, especially as compared to the growth hormone. So the growth hormone is here, and so they're saying, okay, calculate the rate of decrease, percent decrease per minute in the detected level of phosphorylated staph from 10 to 30 minutes. Okay, so looking here, that looks like 10 minutes. This looks like 30 minutes, and if you read the graph, we're going from, about 100, looks like down to about, if I ballpark it, about 38% left, right? So what we have is a 62% decrease, right? And then over 20 minutes, if we wanted to go ahead and divide that, right, by 20, divided by 20 minutes, what we'll get is, I don't have a calculator handy, so 3 point something. Get my calculator here. So 62 divided by 20. Great. Okay, about 3.1% decrease per minute. Okay, great. 3.1 decrease per minute. Okay, cool. Let's look at question D. All right, so the researchers claim that similar regulation of carbohydrate metabolism by hepatocytes when the cells are treated with growth hormone or insulin results from the activation of different signaling pathways. Use data from the researchers' experiment to support this claim. Okay, great. So it looks like when you look at the Y or the vertical axis here, what you should notice is that there's different proteins, right? So this with the growth hormone, it looks like growth hormone activates this protein called STAT5, and the insulin activates a different protein, which is going to be kinase B. So based on this different protein that these different hormones activate, it stands to reason that those are going to be different cell signaling pathways. And if you look here at this, okay, so insulin, high level of phosphorylated protein kinase, yep. And then between 3 and 3.5% per minute, so we did 3.1, close enough, well within the range. And then different signaling proteins are phosphorylated by the two different hormones, which supports the researcher's claim. Okay, all right, so I will post another video very shortly for the second FRQ question in this unit. Okay, thanks a lot.